My talk last week was very much a review type talk. Today, I hope to convince you that the Mauritian Cynomologus macaque is a really good model for studying the role of host MHC genetics in SIV disease course, and also for studying specific questions. They're trying to ask specific questions about T-cell responses to SIV. But today I'm going to present a lot more data than I did last week, data that we've collected in either my lab or when I was part of Dave's lab. So the other thing before I get started is I want to sort of build upon something that Dave talked about last Friday. I think he told you that there are a number of different monkey models that are used to ask specific questions about host immunity to SIV as a model to understand host immunity to HIV. And again, what I hope to convince you today is that this model is a very good one for studying these specific questions about T-cell immunity to SIV as a model for HIV. But I'm not going to sit here and tell you that this is the best model ever for every question, but it does allow us to ask very specific ones. So today I'll briefly talk about how macaques can be used as a model to study T-cell immunity to SIV. Again, a lot of that was discussed last week, so I won't go into that too much. And I'll tell you many of the unique attributes of Mauritian Cynomologus macaques, and then I'll talk about a couple of specific studies that we've pursued in the last few years, one describing an MHC heterozygous advantage in Mauritian macaques, and then another story describing conditional immune escape from CD8 T-cell responses. So I think you saw a very similar slide last week. Studying HIV in macaques can be simpler than studying HIV in humans. You know, in humans, we don't know the infecting viral sequence, the dose, or the route of infection. Pathogenesis is complex. Data can only be obtained from chronic infection, and usually you only get peripheral immunity, although obviously there are more acute cohorts now than there used to be. With monkeys, we can infect with a known SIV sequence. We know the dose that we infect them with and the route of infection. Pathogenesis can be reproducible. We can get data from acute infection soon after we infect the animals. And we also have access to more mucosal tissues, although as Hendrik showed you, you can get samples from axillary lymph nodes from humans, so you can get some tissue samples. But the one thing that's clearly evident is that humans have very complex host genetics, and in monkeys we can simplify this somewhat, and I hope to share with you an exciting model for that today. As I discussed last week, we have the genetic basis of HIV and SIV elite control has been studied pretty extensively, and I described the science paper from 2010 where they identified the role of SNPs in the host MHC and its effect on control of HIV. And I showed you the same Manhattan plot last week where they identified that 313 genome-wide SNPs were all within the MHC, sort of describing the importance of this set of host genetics in control of HIV and SIV. So what's the simplest way to study the role of host MHC genetics in disease course? Well, please bear with my very simplistic model of how I think about this. In humans have a very diverse set of MHC alleles, shown here by this different set of colored boxes. Each individual, or most individuals, have a unique set of MHC alleles. And people who are infected with HIV are infected largely with a unique set of HIV sequences. So the CD8 T-cell responses among multiple HIV-infected individuals are largely going to be unique to that individual. To try to study CD8 T-cell responses in macaques has been simplified greatly by the use of Indian rhesus macaques, and a lot of this work was pioneered by David Watkins at the University of Wisconsin. And although Indian rhesus macaques do have diverse set of MHC alleles, just like humans, you can identify animals who share a single MHC class 1 allele, and that's shown here with a single black box between my two monkeys. I have a very simplistic model of monkeys and humans in my slides too, so bear with that. However, what they did was they infected monkeys with clonal SIV viruses, so the infecting viral sequence was exactly the same in these monkeys. And if you had animals who had the same MHC alleles and infected them with the same SIV virus, you would know at least some of the T-cell responses that would likely be made in these different animals. But again, these macaques can only share one, maybe two MHC alleles. So we wanted to know, can we simplify this model even further? Can we make an even more well-controlled model for studying host MHC genetics and SIV infection? So in about 2005, Dave realized that there were some Cynomologus macaques that live on the Indian Ocean island of Mauritius, which is off the eastern coast of Madagascar. Monkeys were brought to this island about 500 years ago, and then monkeys do what monkeys do. And after 500 years, we have now what is probably the closest thing to an inbred mouse, but now in the monkey model that we can study. So the MHC alleles in these macaques have been identified, both class 1 and class 2. And I keep hitting a button, I guess. We found that nearly all of their MHC alleles can be explained by 7 common haplotypes, which we call M1 through M7. And some of these haplotypes are more common than others. We can find animals who are MHC identical to one another and animals who are MHC distinct from one another. And throughout the remainder of my talk today, I'm going to be showing you these very simplistic models of monkeys, and the little vests below the monkey faces represent the MHC genotypes of these different animals. So an animal with 2 blue vests is an M3 homozygote, and an animal with 2 black vests is an M1 homozygote, and so on. And just to remind you, when I'm referring to a haplotype, that is the entire set of HLA alleles or MHC alleles that come from one chromosome. And if you remember this slide from last week, you inherit 1 haplotype from mom and 1 haplotype from dad during when you have kids. Good enough. So if we move on to describe some of the number of known MHC class 1 alleles in different species, in humans, as I mentioned last week, there are quite a few MHC class 1 alleles. If you added up the number that I put on my slide last week, it's 5,399, quite a few. In Indian rhesus macaques, again, the next best species to study, they have about 1,188 MHC class 1 alleles. But in Mauritian Cynomologus macaques, there are only 69 MHC class 1 alleles. So there are many fewer alleles that we have to think about when we're trying to study this population of macaques. So if we go back to my very simple model of host MHC genetics and infecting SIV viruses, you can see that we have actually simplified this model even further, because we can find animals who have the exact same host MHC genetics and infect them with the exact same SIV virus, so that we know which peptides are likely going to be presented to CD8 T cells in each of our animals. So how does this model facilitate the studying of T cell responses? As I said, we can select MHC identical Mauritian macaques and MHC distinct Mauritian macaques. If we just pick up 100 monkeys off the island and screen them for MHC genotypes, we can find that about four animals are going to have the M1, are going to be homozygous for the M1 haplotype. About 4 will be homozygous for the M3 haplotype. About 5 will be heterozygous for the M1, M3 haplotype, and so on. So we can actually find animals with reasonable frequency for various studies to ask specific questions. Among these monkeys, they share some class 1A alleles. So the 3 most common haplotypes, the M1, M2, and M3 haplotypes, all have the same class 1A alleles. So we often refer to these as universal class 1A alleles, because if you look at most of the Mauritian cyno population, about 90% of the animals actually have 1 copy of one of these haplotypes. So that means that in about 90% of the animals, they will have these class 1A alleles, and they will present those peptides to T cells. So if you're interested in a specific CD8 T cell response that's targeting one of the peptides restricted by one of these class 1A alleles, you can study it in 90% of these monkeys. The last major advantage is that we can map CD8 T cell responses pretty rapidly in these animals, and these T cell responses are quite reproducible among animals. And just to give you an idea, if you look at the M3, M8 haplotype, we've already mapped 12 CD8 T cell epitopes that I'm showing here on this slide, and I know of a couple of others that are restricted by alleles present on this haplotype as well, including 1, 2, 3, 4, 5 epitopes that are restricted by the universal alleles. So these 5 epitopes would be presented by MHC molecules in 90% of Mauritian cynos. And we could study T cell responses to those 5 epitopes in 90% of animals. So how can we use this model to study SIV disease course? Well, we can use this model to look and see whether MHC genotype affects SIV viral loads. Clearly, if we have MHC-defined animals, we can ask these types of questions. What's really interesting is we have the most well-controlled model for studying SIV infection. We can control both for host MHC genetics. We can control for both infecting viral sequence. Do MHC-identical animals infected with the same SIV virus have identical outcomes? So what is the role of host MHC genetics and infecting viral sequence in determining the outcome? And lastly, is MHC heterozygosity linked to a good outcome? And I'm going to go into great detail about the role of MHC heterozygosity in disease course in a little bit. So first, how does MHC genotype affect viral loads? As I said, we have a number of different, we have 7 different MHC haplotypes in our animals. We've infected a bunch of different animals with different genotypes at the University of Wisconsin, and we've measured SIV viral loads over time. And although we don't have too many major conclusions yet from the animals we've studied, it does appear like there are some trends that are beginning to emerge. The different colors, again, represent the different MHC haplotypes. You can see that some haplotypes tend to have lower viral loads than others, where the M1s have lower viral loads than the M5s. But for some of these genotypes, our N is quite small. The most exciting haplotype is obviously the one down here, the M7 haplotype. But we only have about 3 animals with this haplotype so far that we've studied. So we are beginning to make these sorts of correlations with respect to which haplotype is associated with control, and I think that more of these analyses will be performed in the future, but it's clearly something we can ask with this model. As I said before, we have a very nicely well-controlled model where we can look at the reproducibility of SIV disease course and SIV viral loads in MHC-identical animals infected with the same SIV virus. So we can take multiple animals who are MHC-identical, and I'm going to show you data from multiple M3 homozygous animals, and infect them with the same SIV virus. And as you can see here, 10 different animals were infected with the same SIV virus. These animals had the same MHC genotype, and we have completely different outcomes from these animals. And you'll see more examples of this in other data that I present today. In fact, these animals do quite poorly, but we've still got one animal who became an elite controller with this MHC genotype. And we still don't understand why we have MHC-identical animals who have such differing outcomes. But this really provides us with a platform in which we can explore other host genes and other factors that may be involved in determining outcome. So the third point I wanted to talk about is the role, or a study where we've looked at the linkage between MHC heterozygosity and disease course. So we wanted to ask, is MHC heterozygosity linked to a good outcome? And we have a unique group of animals in which we can examine this question. Remember, we have animals who are homozygous and then animals who are heterozygous for those exact same alleles that are present in each of the different homozygous counterparts. And we can compare disease course in animals who are homozygous and heterozygous for the same MHC alleles. As I talked about last week, in humans, a heterozygous advantage has been detected or has been associated with control of HIV infection, such that humans who are homozygous in the HLA tend to progress to disease more rapidly than those who are heterozygous in the HLA. And after this study, the question was raised, is this because these humans who are heterozygous make more CD8 T cell responses, or do they simply have a rare CD8 T cell response that is very effective against HIV? So we've actually performed 2 MHC heterozygous advantage studies, and I'm first going to talk briefly about version 1.0, which we published in 2010. Here, we did a retrospective analysis of animals who were homozygous and MHC heterozygous, and we looked at their viral loads over the course of time. What I'm showing you now is the viral loads for the different cohorts, and in the solid black line is the mean viral load for the homozygous group, and the shaded area is the 95% confidence interval. And in the dark pink line is the mean viral load for the heterozygous group, and in the shaded area is the 95% confidence interval. And what you can see is that the homozygous group had a viral load set point that was about 80-fold greater than the heterozygous group. And if you notice, our cohort was enriched for animals who were homozygous and heterozygous for the M3 haplotype. And in the light blue line is the mean viral load for the M3 homozygous group. And in the dark blue line is the mean viral load for the heterozygous group. And you can see that this difference was nearly 300-fold when we looked at just M3 containing animals. So it seems as though the Mauritian macaques are a fantastic model for being able to explore the MHC heterozygous advantage to control SIV replication as a model for trying to understand the role of HLA heterozygosity in control of HIV viral loads. So we then moved on to MHC heterozygous advantage 2.0, which is more of a prospective study, where we started with 6 M1 homozygous animals and 6 M3 homozygous animals, and then 6 M1, M3 heterozygous animals. So the M1 homozygous animals have the potential to present peptides restricted by all the M1 alleles, and the M3 homozygous animals have the potential to present peptides restricted by all the M3 alleles. And obviously, the heterozygous animals have the potential to present peptides restricted by both the M1 and the M3 alleles. So we could specifically ask the question that I posed to you earlier: do the heterozygous animals have an increased number of T cell responses because they have the potential to present a greater diversity of CD8 or of peptides to CD8 T cells? Or is it simply that the heterozygous animals have an increased likelihood to get the right response? So we started this study in 2011, and we've just been finishing this up recently, and Dave's lab has still been finishing it up as well. If we look, first of all, at the SIV viral loads in these different groups throughout the course of infection, here's what we find. During acute infection, animals who are homozygous for the M1 haplotype shown in black, homozygous for the M3 haplotype shown in blue, and heterozygous for the M1 and M3 haplotype shown in orange are very similar during acute infection. We can get to 12 weeks after infection, and you see that the M1 homozygotes and M1, M3 heterozygotes begin to have lower viral loads than the M3 homozygotes. And this is even more evident at 21 weeks after infection and 52 weeks after infection. And interestingly, you see that same phenomenon that I mentioned to you before, where we have animals who are MHC identical to one another who are not exhibiting the same viral loads. You have a group of M1 homozygotes with low viral loads and a group of M1 homozygotes with high viral loads. And a group of M1, M3 heterozygotes with low viral loads and a group with high viral loads. But if you look at the mean across the entire group, it appears as though the M1 containing animals, whether they're M1 homozygotes or M1 heterozygotes, tend to have lower viral loads than the M3 homozygotes. So again, we wanted to ask this question: is SIV control in the M1 containing animals a function of increased breadth of CD8 T cell responses or a specific response that these animals are making. So Melissa, Buddy, and Dave's lab looked at the number of CD8 T cell responses. So we didn't look at CD4 T cell responses, we only looked at CD8 T cell responses. And we wanted to quantify the number of T cell responses made in these animals a year after infection. And she included the 18 animals I showed you in this specific heterozygous advantage study, as well as some other animals we have at Wisconsin. And if you look at all Mauritian cynos that we've been studying, controllers, non-controllers, heterozygotes, homozygotes, they all seem to have about 5 CD8 T cell responses at a year after infection. So there's no obvious difference in the number of CD8 T cell responses made in these animals at this late time point. And this clearly is the same sort of data that Hendrik showed you guys about an hour ago in his study. But when she found that a specific region in NEF seemed to be targeted strongly by some of the controllers, so she looked at the peptides spanning NEF and tried to measure by interferon gamma ELISpot the magnitude of T cell responses targeting these different epitopes. And if you look here in this one peptide, NEF 193 to 203, the controllers were making a much more robust T cell response to this peptide than the progressors at about a year after infection. So these animals were able to strongly maintain this robust T cell response against this epitope a year after infection. Surprisingly, this T cell response targets an epitope that's restricted by one of those universal alleles. So here you can see simply a mapping analysis, or an analysis where she mapped this T cell response. And on the far right is data from cells presenting the universal allele A1063, which is present in both the M1 and M3 haplotype. So all the animals in that 18 animal study have the potential to target this specific epitope. Yet only some of them seem to be mounting a robust response at this late time point, whereas others are not. And only the controllers are mounting that robust response, whereas the progressors are not. So she then looked at variation within this epitope at 12 weeks after infection, so at a very early time point relative to the time point when she measured the T cell response. And here you can see the frequency of wild-type HW8 sequences, that's the epitope in NEF, in all Mauritian cynos at 12 weeks in this cohort of 18 animals, in the controllers, and in the progressors. And what she found was that nearly 100% of the animals who became controllers had wild-type HW8 sequences at this early time point, whereas most of the progressors seem to have variants that had accumulated at this time point. So it suggests that maintaining this epitope intact is important for maintaining that durable T cell response up until a later time point. So to summarize what we found with respect to the MHC heterozygous advantage, we've detected an MHC heterozygous advantage in SIV-infected Mauritian macaques, which has allowed us to explore this mechanism of heterozygous advantage in SIV-infected monkeys as a model for understanding heterozygous advantage in HIV-infected humans. We found that T cell breadth did not correlate with outcome. However, it seemed that a specific response targeting this region in NEF was important, and preserving the wild-type HW8 sequences has correlated with a better outcome. Unfortunately, though, we still don't know why some MHC-identical animals can mount the HW8 response control while others don't. If all animals can make this response, why are some controlling and others don't? And that's going to be something that we have to follow up moving forward. So the last thing I want to talk to you about is using this model, and actually the heterozygous advantage model that I've shared with you in the last several slides, to answer the question, is there an order to the timing of immune escape? So I just showed you right now that HW8 does not necessarily escape in all the animals. So one thing we can address in these animals is, does escape have to happen in epitope A, and then B, and then C, and then D, and so on? Is there an order? So if you have a certain set of MHC alleles, and you're infected with a certain SIV virus, can you say escape has to happen from epitope A, then B, then C? But if you have a different set of alleles, is it A, and then D, and then F, and whatever it may be? And we can address that question in this unique cohort that I've just shared with you so far. There are 3 acute CD8 T cell responses I'm going to focus on for the next several slides. One that's in GAG, one that's in REV, and one that's in NEF. Again, using the same homozygous and heterozygous cohort that I've just shared with you, all the animals in that cohort would make T cell responses to an epitope in GAG that I'll call GW9 and an epitope in NEF called RM9. But only the M3 containing animals would make a T cell response to this epitope in REV that we call SP10. And we can use this model to try to understand whether escape has to happen in A, and then B, and then C, and so on. So we first wanted to see whether these animals were making T cell responses to these different epitopes as we would predict. And you can see here that the animals are all making T cell responses to the NEF RM9 epitope and to the GAG GW9 epitope as well, but only the M3 containing animals, shown again in orange as the heterozygotes and blue as the M3 homozygotes, are making T cell responses against the SP10 epitope. And we've been able to verify this data with tetramer assays as well. So we then, again, wanted to ask if variants would accumulate in epitope A before they accumulated epitope B. And this is data we've collected by using Roche 454 pyrosequencing of SIV viruses at 4 weeks after these animals were infected. So we're looking at acute variation in these 3 different epitopes. And again, just to remind you, all 18 animals will make a T cell response against NEF RM9. And then we sequence the virus at 4 weeks after infection. And we look at the frequency of wild-type sequences and the frequency of variant sequences. So on the upper left, you see this is the wild-type epitope sequence. The dots represent identity to the wild-type, and then the different capital letters represent variants relative to the wild-type sequence. And on the bottom line, I have just highlighted the % variant in each of these different animals. So on the top, you can see that all the M1 homozygous animals escape from or have accumulated variants in the RM9 epitope. All of the heterozygote animals have accumulated variants in the RM9 epitope, and all the M3 homozygous animals accumulated variants in the RM9 epitope. And this, if you didn't figure it out, I used some conditional formatting to highlight the regions of interest, or the sequences of interest. We then look at the SP10 epitope in REV. And remember, the M1 homozygous animals don't make T cell responses against this epitope, so they have no variation within this epitope. All their sequences are wild type. The M1 and M3 heterozygotes and the M3 homozygotes make T cell responses against this epitope. And you can see that variants accumulate in this epitope in both the heterozygous animals and in the M3 homozygous animals. But then we looked at the GAG GW9 epitope. And remember, I said this epitope is restricted by alleles present in all 18 animals. And all 18 animals make T cell responses against this epitope soon after infection. But surprisingly, the M1 animals had accumulated variants. All the M1 animals had accumulated variants within this epitope, but only 2 out of the 12 animals containing the M3 haplotype accumulated variants within this epitope, suggesting to us that the M3 containing animals needed to accumulate variants in that SP10 epitope that I showed you before they could accumulate variants within this GW9 epitope. Because eventually, if we look at 12 weeks after infection, all of the M3 containing animals have accumulated variants in all of these epitopes. So it suggests to us that there does seem to be this sort of hierarchy or this conditional escape where we have to escape from RM9, then SP10, then we can escape from GW9. So to summarize this idea, again, if you look at these different animals, the homozygous groups and the heterozygous groups, they all make T cell responses against one epitope that we call NEF-RM9. They all make T cell responses against GAG-GW9, and only the M3 animals will make T cell responses against an epitope in Rev. But if we then look at immune escape, they'll all accumulate variants in RM9, and then the M1 animal will accumulate variants in GW9, while the M3 containing animals accumulate variants in the Rev epitope, and then eventually the escape can happen in GW9. So while this is a single example of conditional escape in our SIV-infected Mauritian cynos, similar studies have been performed in HIV-infected humans. Obviously, we have a very well-controlled model here where we can specifically look at this timing of immune escape. But I think that there are similar examples that appear during chronic infection. Probably the HW8 epitope in NEF that I shared with you earlier that we think plays an important role of control in the heterozygous advantage study. It wouldn't surprise me if the reason that that sequence is preserved in those animals has something to do with conditional escape. We've also been studying another epitope in an alternate reading frame protein, and that was my shout out to Paul since he was going to come talk about alternate reading frame proteins. And we've been looking at an epitope in one of those as well that we think is affected by the same mechanism. And this may also have some sort of domino effect, whereas if you slow down your escape in GW9, maybe that slows down escape in another epitope and so on and so forth. Such that when your viral load gets when virus levels are controlled, escape's going to happen more slowly. So just a few final thoughts. Mauritian cynos with specific MHC genetics can be chosen for studies, and I hope that today I've at least convinced you that we can identify Mauritian cynos with specific genetics and choose them for our studies to ask very specific questions about T cell immunity. There are universal MHC alleles in our monkeys, such that about 90% of Mauritian cynomologous macaques have some of these alleles, and we can study similar T cell responses in these animals. We can readily map T cell responses in Mauritian macaques, and we've done this for SIV. Another one of our collaborators at the University of Wisconsin, Tom Friedrich, is trying to use this model to map T cell responses for influenza, and we're interested in doing the same thing for tuberculosis, and I think that the Mauritian macaques could be very valuable for studying T cell responses to a wide variety of pathogens. We can make MHC peptide tetramers, and I didn't show you that data today, but we can generate these with our collaborators in the UK and use them to study specific T cell responses. And again, as I said, we hope that using this Mauritian macaque model may facilitate studying T cell responses to other pathogens as well. So with that, I'll just, I know I'm done a little bit early, but I was encouraged to be done a little early anyway, so that I know it's a little afternoon and you guys are running a little late. Just thank the folks in my lab, Erica, who did a lot of the sequencing that you saw today, and Max, who did some sequence analysis. Dave's lab, who has followed up on the heterozygous advantage work since I left. Tom's lab, who's helped with that as well. Our collaborators in the UK, NCI, and then some funding that I've received. So thanks.